Section 17.4, Disruptions in the Immune System. By the end of this section, you will be able to describe hypersensitivity and define autoimmunity. A functioning immune system is essential for survival, but even the sophisticated cellular and molecular defenses of the mammalian immune response can be defeated by pathogens at virtually every step. In the competition between immune protection and pathogen evasion, pathogens have the advantage of more rapid evolution because of their shorter generation time, large population sizes, and often higher mutation rates. Thus, pathogens have evolved a diverse array of immune escape mechanisms. For instance, Streptococcus pneumoniae, the bacterium that causes pneumonia and meningitis, surrounds itself with a capsule that inhibits phagocytes from engulfing it and displaying antigens to the adaptive immune system. Staphylococcus aureus, the bacterium that can cause skin infections, abscesses, and meningitis, synthesizes a toxin called leukocidin that kills phagocytes after they engulf the bacterium. Other pathogens can also hinder the adaptive immune system. HIV infects helper T cells using their CD4 surface molecules, gradually depleting the number of helper T cells in the body. This inhibits the adaptive immune system's capacity to generate sufficient responses to infection or tumors. As a result, HIV-infected individuals often suffer from infections that would not cause illness in people with healthy immune systems, but which can cause devastating illness to immune-compromised individuals. Figure 17.21. HIV, shown in green, is shown budding from a lymphocyte cell, shown in red, in culture. Inappropriate responses of immune cells and molecules themselves can also disrupt the proper functioning of the entire system, leading to host cell damage that can become fatal. Immunodeficiency. Immunodeficiency is a failure, insufficiency, or delay in the response of the immune system, which may be acquired or inherited. Immunodeficiency can allow pathogens or tumor cells to gain a foothold and replicate or proliferate to high enough levels so that the immune system becomes overwhelmed. Immunodeficiency can be acquired as a result of infection with certain pathogens that attack the cells of the immune system itself, such as HIV, chemical exposure, including certain medical treatments such as chemotherapy, malnutrition, or extreme stress. For instance, radiation exposure can destroy populations of lymphocytes and elevate an individual's susceptibility to infections and cancer. Rarely, primary immunodeficiencies that are present from birth may also occur. For example, severe combined immunodeficiency disease, or SCID, is a condition in which children are born without functioning B or T cells. Hypersensitivities. A maladaptive immune response toward harmless foreign substances or self-antigens that occur after tissue sensitization is termed a hypersensitivity. Types of hypersensitivities include immediate, delayed, and autoimmune. A large proportion of the human population is affected by one or more types of hypersensitivity. Allergies. The immune reaction that results from immediate hypersensitivities, in which an antibody-mediated immune response occurs within minutes of exposure to a usually harmless antigen, is called an allergy. In the United States, 20% of the population exhibits symptoms of allergy or asthma, whereas 55% test positive against one or more allergens. On initial exposure to a potential allergen, an allergic individual synthesizes antibodies through the typical processes of APCs, presenting processed antigen to helper T cells that stimulate B cells to produce the antibodies. The antibody molecules interact with mast cells embedded in connective tissue. This process primes or sensitizes the tissue. On subsequent exposure to the same allergen, antibody molecules on mast cells bind the antigen and stimulate the mast cell to release histamine and other inflammatory chemicals. These chemical mediators then recruit eosinophils, a type of white blood cell, which also appear to be adapted to responding to parasitic worms. Eosinophils release factors that enhance the inflammatory response and the secretions of mast cells. The effects of an allergic reaction range from mild symptoms like sneezing and itchy, watery eyes to more severe or even life-threatening reactions involving intensely itchy welts or hives, airway constriction with severe respiratory distress, and plummeting blood pressure caused by dilating blood vessels and fluid loss from the circulatory system. This extreme reaction, typically in response to an allergen introduced to the circulatory system, is known as anaphylactic shock. Antihistamines are an insufficient counter to anaphylactic shock, and if not treated with epinephrine to counter the blood pressure and breathing effects, this condition can be fatal. Figure 17.22. On first exposure to an allergen, an antibody is synthesized by plasma cells in response to a harmless antigen. The antibodies bind to mast cells, and on secondary exposure, the mast cells release histamines and other modulators that cause the symptoms of allergy. Delayed hypersensitivity is a cell-mediated immune response that takes approximately 1 to 2 days after secondary exposure for a maximal reaction. This type of hypersensitivity involves Th1 cytokine-mediated inflammatory responses and may cause local tissue lesions or contact dermatitis, rash or skin irritation. Delayed hypersensitivity occurs in some individuals in response to contact with certain types of jewelry or cosmetics. Delayed hypersensitivity facilitates the immune response to poison ivy and is also the reason why the skin test for tuberculosis results in a small region of inflammation on individuals who were previously exposed to Mycobacterium tuberculosis, the organism that causes tuberculosis. Autoimmunity. Autoimmunity is a type of hypersensitivity to self-antigens that affects approximately 5% of the population. Most types of autoimmunity involve the humoral immune response. An antibody that inappropriately marks self-components as foreign is termed an autoantibody. In patients with myasthenia gravis, an autoimmune disease, muscle cell receptors that induce contraction in response to acetylcholine are targeted by antibodies. The result is muscle weakness that may include marked difficulty with fine or gross motor functions. In systemic lupus erythematosus, a diffused autoantibody response to the individual's own DNA and proteins results in various systemic diseases. Systemic lupus erythematosus may affect the heart, joints, lungs, skin, kidneys, central nervous system, or other tissues, causing tissue damage through antibody binding, complement recruitment, lysis, and inflammation. Figure 17.23. Systemic lupus erythematosus is characterized by autoimmunity to the individual's own DNA and or proteins, which leads to varied dysfunction of the organs. Autoimmunity can develop with time, and its causes may be rooted in molecular mimicry, a situation in which one molecule is similar enough in shape to another molecule that it binds the same immune receptors. Antibodies and T cell receptors may bind self-antigens that are structurally similar to pathogen antigens. As an example, infection with Streptococcus pyogenes, the bacterium that causes strep throat, may generate antibodies or T cells that react with heart muscle, which has a similar structure to the surface of S. pyogenes. These antibodies can damage heart muscle with autoimmune attacks, leading to rheumatic fever. Insulin-dependent, or type 1, diabetes mellitus arises from a destructive inflammatory Th1 response against insulin-producing cells of the pancreas. Patients with this autoimmunity must be treated with regular insulin injections.